Hi everyone, I am Dr. Sheela, Assistant Professor, PG and Research Department of Microbiology, St.Joseph College of Arts and Science, Autonomous, Cuddalore. Now I discuss about the topic Griffith experiment. First of all, what is nucleic acid? Nucleic acid naturally occurring the chemical compound that is capable of being broken down to yield phosphoric acid, sugar and a mixture of organic bases, purine and pyrimidine. Nucleic acid, the main information carrying the molecule of the cell and by directing the process of protein synthesis. They determine the inherited characteristics of every living things. The two main classes of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid. The following evidence support to the DNA as a genetic material. First one is Griffith experiment, Hershey and Chase experiment. What is Griffith experiment? The Griffith experiment carried out by English bacteriologist Frederick Griffith in 1928 described the transformation of non-pathogenic pneumococcal bacteria into a virulent strain. Griffith combined living non-virulent bacteria with a heat inactivate virulent form in this experiment. He was the first person to discover the transforming principle. Streptococcus pneumonia in several types of strains. Griffith chose two different strains for his experiment. One strain of bacteria has smooth surface and is known as smooth strain, S strain, while the other has rough strain is known as rough strain or strain. Bacteria of the S strain have a smooth surface because they produced a polysaccharide protective coating that forming the outermost layer. Apart from the morphological differences, the Griffith discovered another significant difference between the S and R strains of bacteria. That is, S strains is the virulent strain. R strain is the non-virulent strain that will not cause death in mice. Griffith observed that when he injected the bacteria into mice, the mice infected with the virulent S strain died from pneumonia, whereas the mice injected with the non-virulent R strain survived. During the experiment, Griffith cultured Streptococcus pneumonia bacteria which showed two patterns of growth. One culture plate consisted smooth shiny colony and another one is a rough colony. Griffith infected both S and R strains to maize. The one which was infected with the S strain developed pneumonia and died, while infected with the R strain stayed alive. In the second stage, Griffith heat killed S strain bacteria and injected into mice, but the mice stayed alive. Then he mixed heat killed S strain, live R strain. The mixture was injected into mice and they died. In addition, he found living S strain bacteria dead in mice. Based on the observation, the Griffith concluded that the R strain bacteria had been transferred by the S strain bacteria. The R strain inherited some transforming principle from the heat killed S strain bacteria which made them virulent, and he assumed this transforming principle as a genetic material. Next one is Avery McLeod and M.C. Carley experiment. In this experiment conducted, the ability was not altered by treatment with the enzyme or RNase, but it was completely destroyed by the enzyme. to demonstrate that was substances responsible for genetic informed was a DNA of the donor cell, and hence that DNA as a genetic material. Take one heat-killed estale. The remove the lipids and sugars from a solution of heat-killed estane, protein, RNA and DNA remaining solution. Next, treat the solution with the enzymes to destroy the protein, RNA and DNA, and to add the culture containing living or cells. The observed for transformed by testing for the presence of virulent estane. The transforming require DNA, therefore is the genetic material. It is a DNA as a genetic material. Next, Hershey and Chase experiment. It was performed by Alfred Hershey and Martha Chase in 1952. In this experiment, the E. coli T2 phage was taken as the test agent. This is also called as a blender experiment because a kitchen blender is used as a major piece of experiment. In this experiment, the E. coli T2 phage was taken as the test agent. In T2 phage, DNA is the only phosphorus containing as a genetic material, whereas a capsid shell is enriched with a sulfur containing amino acid like cysteine and methionine. Sulfur is not available in DNA. So the phage was subjected to radioactive labeling with a P32 and S35. Since the phage protein do not contain phosphorus, only DNA would be labeled with a phosphorus 32, while the phage protein are labeled with a S35. Both kinds of labeled phage particles allowed to infect E. coli separately. The phage multiply in E. coli and are released as progeny phases after 20 minutes. When the progeny phages are studied for radioactive activity, it was found that the progeny phage carried label the phosphorus 32, not the sulfur 35. This experiment clearly indicated that only DNA is being injected into bacterial cell. So, DNA as a genetic material to transfer the one generation to another generation of genetic information. Thank you.